As Jim said, I'm a researcher, so you probably will have something to say about my visualization attempts here. But I also should say that the whole field of genomics and epigenomics and the challenges that have come from this have really only popped up in the past few years. And it really occurred to me looking at this previous really excellent talk on cartography, where people have thought about these things probably for hundreds of years. We've been thinking about this in terms of the genome for a little more than 10 years, and in terms of the epigenome, maybe 5 to 7 years. So these are very fresh problems, and there's a lot of data that we're facing right now, and there's a lot of opportunity to really help out and make these data accessible to a wide audience, a broad audience that's not necessarily research focused. So I think this is a great opportunity to get into this and work with new types of data. So I thought of introducing, so what is epigenetics? As I said, it's really come of age in the past few years. And so I went on Google and tried to find a nice cartoon. I found this, and I guess this is probably me right now. And you ask me something, and I'll just say it's due to epigenetics. So that didn't really help. However, I found a very good example, and really one of the poster childs for epigenetics. So Jim already said this. One of the things we're facing in epigenetics is like the big question. So almost all the cells in our bodies have the same DNA, but still they're able to make like skin cells and liver cells and brain cells. So how does this really happen? So the cat has the same problem. All the cells in this cat will have the same DNA, but it will still end up with this different like code color pattern. And so this decision is made in the hair-producing cells early on in development, when a decision from two alleles, so we have 2 copies of each chromosome, a decision is made if the orange allele, the orange gene, will be on versus not. And so the cells that decide to have the orange gene on will produce like orange fur color, while the cells that do not have the orange gene on will express genes that make the fur black. So this is a classic epigenetic problem. But of course, this is not the only place where epigenetics really impacts our lives. In fact, epigenetic regulation is implicated in probably all the processes that revolve around our lives, starting from early development, so the fertilized zygote, to the fully grown and developed organism with all different tissue types, cellular differentiation, so how do we go from one cell with the same DNA to like all these different cell types that we have in the body. X inactivation in females, so all the females in this room have an extra copy of the X chromosome that the males do not have. We need to actually shut down this extra copy of the X chromosome to prevent disturbances in gene expression. So one of these X chromosomes is parked in a silent state, and this occurs through epigenetic regulation. But epigenetic regulation, when it starts to go wrong or start to become less accurate, is also implicated in some of the phenotypes we observe with aging, both cellular and large scale, and also in cancer, which is really what my research, where my research interest lies. So there are different epigenetic mechanisms in the cell has at hand to control these type of state and transition changes. As you probably know, the DNA is packed up in these like compact structures that we call the chromosomes down here. And if we unwrap this, we'll see that the DNA is further wrapped around these protein complexes. They're little spheres called nucleosomes. And these nucleosomes consist of histone proteins. So each of these will contain different histones. And these histones have little tails stick out. So these are like little like antenna signaling posts here. And these antennae can be modified, which is what we refer to as post-translational histone modification. And depending on these little flags or bookmarks that are hanging off this antenna, the DNA lets the cellular machinery know either transcribe me, I'm active, or don't transcribe me because I'm a brain gene and I don't want to be translated in transcribed in a liver cell. This whole thing of DNA with its associated proteins we refer to as chromatin. So I'll be using this term a lot. In addition, there's also direct modification of DNA through attachment of a methyl group to cytosine residues. So this does not involve extra protein that's wrapped around. I'm just going to mention this now. The talk will be focused mostly on histone modifications. So if we zoom in on 2 of these nucleosomes, you can see these individual histone proteins. There are 8 of those 2 tetramers in each of the nucleosomes. And this is the tail I mentioned. These are the different amino acids here. And you can see there are different chemical groups that can be attached to these residues at different positions, such as methyl groups, phosphor groups, or acetyl groups. And depending on what group it is and where it occurs in this tail, the signal can be very different from active or repressed. So all these marks are projected down, by a whole battery and hundreds of enzymes that are specialized to recognize these modifications, to write them, and also to erase them. So this is a cartoon from a recent publication of the lab. You can see that there are lots of different proteins. They form complexes. They perform different functions regarding the actual mark. So you'll see a lot of the K, which stands for lysine residue. Lysine residues are some of the most studied ones. And there can be a specific, so not only to the mark, but also to the genomic feature that they act on. For example, distal elements or promoters or transcribed regions. So with this, we have, with these histone modifications, we have tools at hand to really now bookmark and classify the genome according to its regulatory function. For example, up here, we have the structure of a gene with its promoter and some exon-intron structure. And at active promoters, we have particular histone modifications, such as lysine methylation up here, acetylation, histone variants. In the gene body, when the gene is transcribed, we often observe K36 methylation, K79 methylation. In contrast, when a gene is inactive, such as down here, the promoter and often the whole gene locus will be covered with lysine 9 methylation and lysine 27 methylation, marks that are actually associated with physical compacting of this part of the genome to stow it away in a part where we either don't need it forever or we'll just take it out sometime later when we need it again. So in addition to these well annotated and through years and years and decades of study, identified gene loci in the genome, of which we have tens of thousands, we've also found out that histone marks can mark other functional elements in the genome. A lot of them we call enhancers, and we think they perform functions that where a regulator can be bound by a specific transcription factor, fold into a gene locus, and enhance its transcription. There are other elements we can recognize based on histone modifications or protein bindings, such as insulator elements, which separate different compartments in the genome from each other. So how do we detect these epigenetic marks? What are the experimental strategies that we have at hand? So for protein detection, maybe histones with specific modifications or proteins that are bound to the DNA, we use chip chromatin immunoprecipitation. And the way this works, we take the DNA from a cell, we cross-link the DNA, so all the protein is really tightly bound to the DNA and doesn't come off. We fragment the chromatin through shearing. We immunoprecipitate with a specific antibody. It's really the cool part. We have a specific antibody that will only recognize a modification, let's say, on lysine 4. So if we immunoprecipitate with this antibody, we'll only pull out DNA fragments that actually have this modification, and then we'll throw away the rest. So we purify, amplify the DNA, and the next step is really where the revolution has come in the last few years. We do high throughput sequencing. And so with the vast increase in sequencing technology, but also becoming cheaper and cheaper, we're now accumulating masses and masses of data through this experiment. So what do we do with the reads that come out of this sequencer? We visualize them as fragment count per genomic position. So how exactly does this work? These are DNA reads that we get from the sequencer. They're typically short. We have the reference genome available. It's actually really become something that we don't even think about, but the reference genome in its current shape has really only been around for 12 years. So this is really like a new field to get into. So We count the reads at each position. So you can see at this position we had 1 read. At this position we had 2. These 2 overlap. And then we have 3 reads here. And this position we have 4. And then we can generate these density maps, basically just showing the counts of reads at each position. And so whenever you see a peak in this track here, indicates that there's accumulation of reads, which again translates back into the protein being bound at this particular place in the DNA in the original cell that we started with. So with this, we can now annotate the little cartoon I showed earlier. So this is the gene locus. This is the promoter and the gene body. So at a transcribed gene, we'll have a K4 trimethylation signal. You can see a very nice peak here. And as I said before, in the gene body, we'll observe K36 trimethylation. So you can see blue signal going all the way across this gene body. Down here is a silent gene. You can see that K4 trimethylation is absent, as is K36 trimethylation. Instead, we have K27 trimethylation covering this whole locus, really indicating this gene is packed away somewhere in the cell where we don't need it. I also told you that we have these enhancer elements. We have ways to recognize them based on their chromatin patterns. Active enhancers are marked with K4 monomethylation, as well as K27 acetylation here. They're inactive, or what we call poised enhancers, if they only have K4 monomethylation, but they lack K27 acetylation. We think these are sitting there probably ready for activation at some later time point. But most of the time, actually, there's no mark if this regulatory element is not being used in the cell. We just can't tell because it's not marked by anything. All right, so these are just a few experiments in exactly 6 cell types that we profiled. A single gene locus, you can see that this becomes complex very quickly as you do lots of marks and lots of cell types. In staying in line with the change detection, you can probably appreciate that here these different cell types are very similar. Here they're different, but it's actually very difficult to pick this out once you increase cell numbers and increase experiment types. So what do we actually want to do with this information? So we gather all these sort of linear maps, lots of histone modifications, can tell us lots of things. We can combine these with transcriptome annotation, gene expression data of different types, and then really go from this linear map where we have expression of factors, they go bind somewhere else in the DNA where we can see them, where they are next to genes, they probably regulate. So from this linear representation, try to reconstruct regulatory interactions. That's really where my interest is, in detecting these regulatory interactions in brain tumors. And in particular here, another thing we've really just kind of learned of epigenetics is that tumors are probably not homogeneous masses of cells. So tumors are more like communities of different cell types, and the cells in the tumor will have different functions by themselves and different properties. So this is one of the prevalent models in the cancer community right now. It's also referred to as the cancer stem cell model because in tumor we have different cell populations indicated here by different colors, and only this pink population in this example is able to seed tumors if we inject these different cell populations into mice. The other cells are not tumorigenic. And if you keep doing this, you'll find that this sort of similar same population will continue to form tumors in a serial mouse experiment. And so people think that these probably are the really critical cells in a tumor that propagate the tumor aggressiveness phenotype. So this heterogeneity in tumors is being well studied at the moment, and people have already demonstrated that tumors can be genetically different. So their DNA actually varies. Tumors have very unstable genomes, so they go through different rearrangement events, and then these individual populations grow out. So tumors are genetically heterogeneous in many cases, but they can also be epigenetically different. So same DNA, but different properties, just like a skin cell can be different from a liver cell. We use a very specific system to study the epigenetic differences in brain tumors. So this is an image from Wikipedia. You can see there's a brain tumor. The impact of this disease obviously is large, and we're interested in finding ways, of course, to cure and prevent this disease. In our particular model, we have cells from patients that were operated on at Mass General Hospital, and we culture different cell populations, either in serum-free media, these are the things that we refer to as the stem cells, and in serum-supplied media. Serum-supplied media is traditional cell culture media, it's been used for decades to study normal cells as well as transformed cells. We can identify these cells by morphology, so you can see the serum-grown stem cells, they grow as these spheres. These serum-supplied brain cancer cells, they grow as these elongated fibroblast, very differentiated cell type cultures. They express different markers, so we can also separate them by their marker expression. This is a fact. So you can see that in the stem cells, this population is very large, so lots of cells express this particular marker. In this example, hardly any of the cells express this marker. But most importantly, and most interestingly to us, is that if we take these two populations and inject them into mice to measure their tumor initiation capacity and their aggressiveness, the cells grown in this traditional media here and the differentiated phenotypes, they don't actually form tumors, or only if we inject really, really large numbers of cells. However, the stem cells are really efficient at forming tumors, and we have to sacrifice the mice pretty rapidly. So we're really interested in the question, I'm sorry, you probably can't really see this there, but what are the epigenetic determinants of these two different cell populations? I should tell you that from what we've seen on a large scale, these are genetically not different. Okay, so they have typical cancer aberrations, such as deletion of tumor suppressor genes and amplification of oncogenes. So these large-scale effects are not what produces this difference. So it's purely, or we think it's largely epigenetic. So we did this experiment at ChIP-seq, as I told you before. This is the K27 acetylation mark. Actually, same locus I showed you before, and you can see here that we have a lot of activity with this active mark here in the cancer stem cells, but not in the differentiated serum-grown cells or in normal brain cells that we used for comparison. So this is pretty nice to see. We're particularly interested in this locus because this gene had been implicated in brain cancer. So it's very nice to use the IGV to look at these things if we know what we're looking for, and it displays very well that there's a difference in the cancer stem cells compared to serum-grown cells. However, the genome is pretty large, and there are lots and lots of these things. In fact, there are like 50,000 of these individual peaks, and we have to go in and really make sense of this large scale. And not only this, but we also have to visualize how the differences play out here. And so, yeah, these are elements you to be looking for. So one way to do this is just take all these things, put them on their side, and display them as a heat map. So the center of the peak now is in the center of this line here, and you can see there are different size peaks. So some of them are a little wider, and then they trail off as they were sorted. But more importantly, this allows us to find and identify different classes of these elements in our experiment. For example, these here, you can see, are both present in the stem cells as well as in the serum-grown cells. Well, this set is only present in stem cells, and this set down here is only present in the serum-grown cells. So after we've identified 16,000 of these elements here, we can do some science with this. And in fact, it is known, it's known that at these enhancer sites, transcription factors that can be specific to a particular cell type go and bind at these enhancer sites. And so we can go and look for the DNA under these sites that we've identified and look for motifs, for recognition motifs for transcription factors that are overrepresented here. So we did this, found different classes of motifs, which is very reassuring. And then went off to actually do the same ChIP-seq experiment for 4 of these transcription factors. So here you can see how do you really go and bind at these K27 acetyl regulatory sites here. In this case, all 4 actually bound at the same site, which is pretty cool. Again, this is the single locus view. we wanted to get a better idea of what's going on large scale. So here you see the same sets I just showed you before. Then you can see how these 4 transcription factors bind at these K27 acetyl regulatory sites, but don't actually go and bind at sites that are inactive or not primed for this particular cell type. So I should say that these transcription factors are all ChIP-seqed in the cancer stem cells. So that makes sense since these elements here are not active in the stem cells. Another way to represent this and to look at some behavior actually of transcription factor binding is to just sum up this whole map into a single graph here. And if we do this, we can see, as I showed before, actually that this K27 makes this pattern that's bimodal here. So with actually a dip in the center of the peak. And it's not surprising because if you look at transcription factor binding now, you'll see that the transcription factors actually tend to bind exactly in this dip. So the way this occurs in cells, the transcription factor binds and is flanked by nucleosomes that are marked with this K27 acetyl mark. All right. So how do we go from this really linear and small-scale map to something that's more integrated and probably 3D? We believe that sites that are near genes and are bound are likely regulators of this gene. So if we assume that these regulatory sites here near this gene actually regulate this gene, we can start counting genes and their regulatory factors in this fashion that I showed before. So again, going from linear transcription factor being transcribed, binds to a site near another gene. Imagine this is what happens in real life. We can go from this to actually make networks. So these are the 4 transcription factors I introduced before. You can see here that each of these transcription factors has their own cloud of target genes. So here are, in gray, are little target genes. You can see a little more connectivity here. So there are genes that are not only regulated by a single factor, but there are also genes that are regulated by all 3 or 4 factors. And if we lay expression data information on top of this, we can also infer if these regulatory interactions are activating or repressing. So in this case, I've added correlation with gene expression. And so if you see a red edge, the inferred regulatory activity is positive on this target gene versus a blue edge, in which case the regulatory activity is negative. This is just a very small study of my own research. We have very limited amount of cell types. Really, we have 2 classes, different cell types. If we add in the normal brain, that makes it 3, but it's a very limited study. And we really focus on 1 mark or maybe a few more. However, if you start looking at this in more large-scale perspective, then you really start seeing patterns that occur in the cells in our body. And so there have been different efforts to do this. One of them, the lab is involved in is the Roadmap Epigenomics project. The Roadmap Epigenomics project profiles lots of primary human tissues. So there's all types of cells from different developmental stages and different parts of the body. In this paper that just came out, the lab looked at K4 monomethylation sites, again, another enhancer site. And if you classify these by their cell types here, you start seeing patterns. And if you look at these patterns here and do the same transcription factor analysis, you'll find that, in this example here, we have enhancers that are specific to embryonic stem cells up here, and that they also enrich for motifs for OCT4 and SOX2, probably the most known stem cell factors in the field at the moment. And you can see how each of these different cell type groups will have their own specific regulatory sites. Now, this is, again, just looking at 1 mark. And if you recall the picture I showed you with the lots of tracks, it becomes very difficult to process all the combinations of what's going on, active promoter, transcript, regulatory element, repression. So the Encode project in the paper that was published in 2011 this was a collaboration with Manolis Kell's lab at MIT. They really looked at all the chromatin marks and came up with combinations that are indicative of particular functional states. So in this case, you can see that an active promoter has very strong signal for K4 dimethylation, K4 trimethylation, K27 acetylation, as well as K9 acetylation. A strong enhancer also has these marks, and in addition, K4 monomethylation. And you can see that, like in repressed genes here, there's only K27ME3, and these marks here correspond to transcriptional elongation. So instead of going from lots of marks, you can sort of reduce this into functional states by annotating them. And if you do this, you can start looking at many cell types now with a summarized chromatin state. In this example here, we have a gene that's marked with active promoter in pretty much all the cell types here, and you can see transcription. And you can start identifying things that are different, as in this example where this gene is active only in the bottom half of the cell types here, but it's repressed in the top half. All right, so I hope I've sort of got you a little bit excited about the new and rising field of epigenetics and epigenetic data. And as you can probably see, there's a lot of visual challenges that are facing us now and in the future as we accumulate more and more and more data. I just gave you a few simple examples of representation that we have at a single locus, the classical ChIP-seq track map, which of course is limited through space, both in terms of the amount of genome we can look at, but also the amount of tracks and cell types we can look at. I showed you some representations of many loci and few cell types. Yes, you can expand this, but of course, this is also finite space. This is an example of many loci and many cell types, but only a single mark. And I've shown you an example of many marks and many cell types. And if you have ideas of what to do more, really, this is the place and time to do it. So I just want to thank all my collaborators here and funding, and thank you for your attention. If you have any questions, I'll take them now.